Dr. Hameroff, why don't you take it away?
Thank you, Allison. Thank you all for being here. I'm going to talk about anesthesia and consciousness. And what you see in the slide is we'll get back to, is ether frolics. At low concentrations, anesthetics, gas anesthetics, have a euphoric, giddy effect, and then you breathe more and you go to sleep. This is a biphasic effect seen in a lot of types of drugs. Succinylcholine, for example, causes every muscle to twitch and then relax. So it's a common feature of many drugs, but anesthetics, the dependent variable. So anesthesia and consciousness. So in the year 2000, the millennium, this book appeared about the greatest inventions of the past 2,000 years. And as you can see, anesthesia is at the top of the list, alphabetically at least. And I wrote this chapter, and what I said was, imagine we lived in a world without anesthesia and we faced, breaking an arm or having an appendix burst or everything else. We'd live in fear. So having anesthesia really makes a difference. In ancient times, before anesthesia, in Assyria, Egypt, Greece, China, India, they used drugs like cannabis, mandragora, which is a nightshade, opium, belladonna, alcohol, and dozens of medicinal plant alkaloids. The Assyrians, Egyptians, and Greeks induced artificial sleep by pressing both carotid arteries. This is frowned on today. And Peruvian shamans actually did local anesthesia with coca leaves. They would chew the coca leaves and then spit into the wound for trephination. So this was maybe the first local anesthetic. So here you can see some of their work. They actually did a pretty nice job drilling holes into the skull to let out evil humors. How that worked out clinically, we're not sure. But here is a picture of a trephination with the shaman having just spat into the wound. And I assume they had a timeout and they're now proceeding with the case. We don't know how it turned out. In 1540, sweet oil of vitriol containing diethyl ether was synthesized and used as a supplement in chicken feed, as its sweet taste made the chickens eat more and fatten up. But the chickens tended to then fall asleep. In the 19th century, gases diethyl ether, nitrous oxide, laughing gas, and chloroform were found to be euphoric when inhaled at low concentrations and were used socially. And here are the ether frolics that I mentioned before, showing the biphasic effect. Low concentrations, they cause giddiness and euphoria, and then you keep breathing and you fall asleep. And here, nitrous oxide was also used, laughing gas. And you can see at the top, it says living made easy. And at the bottom, probably a sexist remark, prescription for scolding wives. And then we move along to the ether dome and the Morton's demonstration of diethyl ether. And then you can see some of his colleagues. There's Dr. Lichtenthal, there's me, there's Dr. Leslie, and a few others of us, or just kidding. Here's another sort of famous case that happened at our hospital in the mid-'80s. Jack Copeland, the heart surgeon in the middle, had been doing heart transplants, and he did the first bridge to transplant. And this Jarvik 7 was put into a patient who was dying of cardiomyopathy. There was no heart available. And the guy lived on it about a month, and then a heart came in one night, and he put it in. This was, I think, the first, pardon me, bridge to transplant. And you can see in the background, Dave Wiggum, one of our really great residents, and myself and Randy Cork, who's now in New Zealand. Dave is in Wenatchee, Washington. So let's talk about anesthesia. What is anesthesia? Immobility, no movement in response to surgical painful stimulus. Amnesia, no memory of events during anesthesia. Loss of consciousness, no awareness during anesthesia. However, about 25 years ago, the loss of consciousness began to be omitted from the definition of anesthesia in a paper, for example, by Ted Eager and his colleagues, UCSF and the West Coast, and then Campagna et al., including Keith Miller from Harvard, on the East Coast, and kind of the West Coast Mafia and the East Coast Mafia decided to get rid of loss of consciousness from the definition of anesthesia because they said that consciousness cannot be measured, observed, or explained, and or could be illusory. And there are many philosophers and neuroscientists who claim that consciousness is an illusion of whom to whom is unclear. But and I'll come to why people think that, because it's difficult to explain. This omission of a loss of consciousness from anesthesia was followed by what I would call an era of great awakening. Here's an article from the Associated Press, 2008, 30,000 people a year awake during surgery. And this commented on an article about comparing the BIS monitor to just following clinical signs, which is what we do when we don't have a BIS or a SedLine. And there were actually more cases of awareness in the BIS study. This was a couple thousand patients in each group from WashU St. Louis, I think. And they commented that 60 million per year were spent on BIS electrodes. And I'll just leave it at that. They didn't prove to be any better. And there's a couple of problems. Number one, the BIS and the SedLine tell you what happened a little while in the past, and we want to know what's happening immediately. And they don't really tell us what that number means. So it's a little bit annoying for that. But I don't use them. And I find when I do use them, I wind up staring at the number and ignoring the patient, which is the opposite of what we should be doing. Then there are presently still consciousness deniers, I would call them. For example, Emory Brown and his group from Harvard or MIT. And in this paper, they discuss 5 classes of receptors affecting arousal and sedation. And they talk about GABA receptors and drugs like propofol, opioid receptors, drugs like fentanyl, et cetera, NMDA, ketamine, alpha adrenergic, dexmedetomidine, and dopamine neuroleptic. And they do not discuss consciousness nor gas anesthetics, but conclude our analysis shows that general anesthesia is less mysterious than currently believed. So if you ignore consciousness, yes, anesthesia is fairly easy to explain, but that's kind of missing the point, I would say. So let's talk about MAC. Eager did a really great thing with Larry Seidman and others coming out with this idea of MAC, which is essentially an ED50 for anesthesia. Now, all other drugs that we use, most other drugs, are intravenous, polar, soluble, and anesthetics are completely different. And most people come into this from pharmacology to study how anesthetics work. So this was actually a great step forward to define an ED50 in terms of gas phase. So it's the equilibrated concentration of inhaled gas at which 50% of animals or humans move or don't move in response to a surgical stimulation. So it's inversely related to potency. So in the animal studies, it was usually clamping a tail, something like that. And this was the seminal paper in 1965. So immobility, MAC addresses immobility. There are other varieties of MAC. At the top, we see MAC for movement, one MAC. Then MAC awake, for consciousness, 0.2 to 0.5. Memory, about the same, 0.2 to 0.5. And then to prevent autonomic response, 1.5 to 2.0. So this refers to suppressing hemodynamic responses to surgical stimulation. So they're prepping for 5 minutes, the patient's equilibrated, they take the knife and the patient moves. So if you're at 1.5 to 2.0 MAC, you'll suppress that. Of course, at that range, we may also get into causing our own suppression from the anesthetic, but this refers to, MAC bar refers to preventing responses to surgical stimulation. So if we put these all on one line, we can see that in the middle is MAC. They're all bell-shaped curves, moving with stimulation. To the right is MAC bar, preventing or blunting autonomic responses. And on the left is MAC awake and also MAC remember, which is based mostly on response to command and amnesia, and to some extent, loss of global coherent EEG and consciousness. Probably the best study EEG goes way back to Eroy John at NYU. who studied many patients going to sleep and emerging and came up with that. It's been repeated in many ways since. So MAC for MAC, immobility, where is it mediated? In the early 90s, Antonini at UC Davis measured MAC in goats. And then he sequentially severed the cortex, measured MAC, severed thalamus, measured MAC, and the brainstem. MAC did not change. And he concluded that spinal cord reflexes mediate MAC. Now, this is a little bit deflating to us because we think we're treating the brain, and we are. But the movement per se, which happens at 1 MAC, is one thing. This doesn't speak to the MAC consciousness, which happens 0.2 to 0.5, as I said. The other conclusion from this is that movement or autonomic response above 0.5 MAC, above a half a MAC, is very likely to be non-conscious. So that's why, in the patient's hypotensive, we make sure they have a half a MAC and they should be okay. Also, non-conscious movement or autonomic response is arousal. It doesn't mean they're awake if they move. So if we put arousal there in the middle, that's more accurate. So loss of consciousness, I'm going to skip over memory because it's more difficult to measure, and I have my own thoughts about that. But it's difficult to determine, and often described to response to command. For example, open your eyes, squeeze my hand, take a deep breath, scratch your eyes, don't scratch your eyes. So if they respond to command appropriately, we assume they're conscious and awake. And based on this, MAC awake is thought to be 0.2 to 0.5. So here we have it where we've kind of zoomed in on the MAC awake 0.2, or actually less than 0.5. And the thought is that response to command upon emergence is asserted to imply conscious awareness. Maybe, because it's also possible that you can be in this range or even higher and respond appropriately, but be non-conscious, what's called in philosophy zombie behavior. A zombie is someone who looks and acts like us, but actually has no inner light, no inner consciousness. So, and when a patient first wakes up and they kind of stare at you, I often wonder, are they really conscious or what? So they could be responding in zombie mode. Similarly, they may be conscious but don't respond. They could be playing possum, and maybe you're saying take a deep breath, and they're thinking, go away, I'm enjoying my opiate, and I feel good for the first time in a long time, something like that. So we don't really know, but roughly 0.2 to 0.5. 0.5 is a good number to remember, even though it may be plus or minus. So how does consciousness occur in the brain? What can we learn from anesthesia about how consciousness occurs in the brain? So in Western philosophy, the world out there is all in our heads. Here, this is Plato, by the way, supposedly. Bing, and I'll use Bing to imply conscious awareness, because remember, the brain is quite active even when it's unconscious. If you do a spine case and do monitoring, or a neuro case, and you're monitoring evoked potentials to the brain, the patient's unconscious. So consciousness is something different, which is what we're trying to figure out. So Bing is meant to imply having conscious experience. In Eastern philosophy, consciousness pervades a deeper level of reality. Being is everywhere. Eastern philosophy would say we're swimming or we're a wave in a sea of consciousness. Maybe that's right. Going back to Western philosophy, Plato realized that because the world out there is all in our head, we don't really know what's out there accurately. Conscious perception may only poorly portray reality. And he came up with this allegory of Plato's cave, where these prisoners were able to only see straight shadows on the cave wall with a fire behind them. And to them, that was their reality, or 2 dimensions were their reality. And this implies to some people that there are deeper levels, deeper dimensions to our own present reality, which may be true. And along these lines, that we can't really trust what's out there based on our consciousness. Descartes said, I think, therefore I am. That's the only thing he knows. And he said that we could each be a mere brain in a vat, fed information by an evil genius. And here's a brain in a vat thinking that he or she is walking outside in the sun. So let's go back to Western philosophy and the brain. And so here's a brain, and we see the thalamus, and sensory inputs, except for smell, route through the thalamus. So here's the thalamus in the middle. So whether it's from the eyes, from the spinal cord, from the ears, from wherever, it goes to thalamus, except for smell, which goes directly into olfactory cortex over here. And from thalamus, the signals go to the appropriate sensory cortex. In the case of vision, this is in the back of the brain, V1. So the green arrow 1 is the thalamocortical projection for vision to the back of the brain. From primary cortex in the back of the brain, there's a second wave through associative cortex that goes to the front of the brain. And this could go through motion, color, meaning, shape are picked up in visual cortex. Then you get to the others, and there's integration and association until you get to the prefrontal cortex and areas around that. And then there's a third wave projecting to other areas of cortex throughout the brain. And it looks like it's this third wave that's mostly associated with consciousness. But some people think thalamocortical projections are conscious, for example, to the back of the brain or to any part of the brain, depending on the sensory cortex. So for example, thalamocortical first wave or first order theories of anesthesia and consciousness include Mike Alkire, who's an anesthesiologist or at least he was at Irvine, and Ned Block, who's a well-known philosopher at NYU. They don't work together. They have similar views coming from anesthesia and philosophy. They suggest that consciousness depends on these large-scale thalamocortical interactions, and that, at least Alkire says, that anesthetics act by blocking thalamocortical projections. So here's some data from one of Alkire's papers, and it's actually kind of a meta-analysis of a number of different studies. So the colors are different, but the colors all indicate decreased blood flow. And this is fMRI studies. And what fMRI and blood flow and bold signal have to do with consciousness is another story. I'll come back to that. But at first glance, this looks pretty convincing, because the thalamus and the cortex are all diminished in terms of cerebral blood flow and activity, with clonidine, propofol, more propofol, sevoflurane, halothane, isoflurane, lorazepam, et cetera, even dexmedetomidine reduce the blood flow and the activity in thalamocortex. So this is an argument on their behalf that consciousness depends on thalamocortical projection, and anesthesia takes that away. So if that were the case, then thalamocortical stimulation might be expected to reverse anesthesia and cause consciousness. And this paper came out last year from University of Wisconsin, which has a great anesthesia department. We had Kelly McQueen here a month or so ago, and has a long story tradition. And this is from Yuri Salmon's lab. And what they did was they had monkeys who were anesthetized with isoflurane at 3 different concentration ranges.
Only do it on Wednesdays.
Low.
For late day. And then
Could somebody mute, please? Thank you.
Oh,
At low, medium, and high concentrations.
There's
And
two.
the MAC of isoflurane
Wally,
is,
can you mute your stuff? Sorry, Stu, to interrupt. Okay, thank you.
thank you. In high concentrations, the MAC of isoflurane is 1.17. So it's near the top of what they call the medium range. They then put electrodes, or they implanted electrodes into the central lateral thalamus. After hours of anesthesia with no stimulation, electricity was applied to the central lateral thalamus. At the low and medium doses, the monkeys responded with arousal and movement. Above 1.2%, no such reaction. They didn't respond. So basically, arousal movement was suppressed at about 1 MAC, pretty much what would be expected. Arousal doesn't necessarily imply consciousness. Nonetheless, they kind of claimed or hinted in the title and throughout the paper that they were reversing anesthesia to cause consciousness. But they don't really know that they cause consciousness. They ablated arousal at 1 MAC, which is what happened, which is what you would expect. So I think this is kind of a bait and switch paper, and we see a lot of these talking about consciousness. When it gets down to it, it's some correlate of consciousness that may or may not reflect consciousness. So beware bait and swapping. Not to beat a dead horse even further, but Ben Libet in 1970s had some very interesting studies, one of which he stimulated the thalamus in humans and looked at their cortical potentials and when they felt it. So he had patients who were undergoing neurosurgery and with their brain under local, with their brains exposed. And he worked with a neurosurgeon, Bertram Feinstein, and he did various studies. And he would ask the patients when they felt it. And in this case, they did not feel it. Thalamic stimulation resulted in evoked potential, which is normally associated with conscious perception and activity, but then it stopped. When he stimulated the hand itself, so this was a thalamic stimulation of the hand area. If he stimulated the hand itself, he got the evoked potential. The patients said they feel it right away, but what he determined was that what was necessary was 500 milliseconds of ongoing activity after the evoked potential to get to what he called neuronal adequacy. And without this 500 milliseconds of activity, so it's absent here, there's no neuronal adequacy and there's no conscious experience. And he concluded that the brain somehow sent information backward in time. And when this came out, he was greeted with derision and boos from neuroscientists and say that's impossible. Well, in physics, it's actually not impossible, but he was berated but he sort of stuck to his guns. And more recently, the physics has shown that this is indeed possible. And I'll come back to that point if I have time. But the point is that thalamic stimulation per se doesn't cause conscious awareness. To further beat this dead horse, psychedelics expand consciousness. Do they increase thalamocortical activity and fMRI signals? So psychedelics cause expanded hallucinations, deeper sense of reality, et cetera, et cetera. So you'd expect the fMRI to kind of light up like a pinball machine. So in this study, at least most people would, and I did until I saw this study. In 2012, Carhart Harris, who was at Imperial College in London and is just now moving to UCSF to start up their new psychedelic research program. He's been to our consciousness conference many times. I had this paper in PNAS, Neural Correlates of the Psychedelic State as Determined by fMRI Studies with Psilocybin. And the blue indicates decreased cerebral blood flow after psilocybin versus after placebo. So it's a subtraction film. So this tells you where there's reduced activity under the influence of psilocybin. These people were in an MRI scanner with an IV getting intravenous psilocybin, and they reported later what they were feeling and thinking. They didn't attempt to report during the experience because of artifact of talking and so forth. But they reported later that they were having profound psychedelic experiences, and you can look it up in this paper. But their fMRI looked cold and dark. And they repeated this with EEG and found the same thing, that EEG was suppressed and almost flatlined. So this was kind of a confounding finding. Excuse me. Conclusion being that psilocybin expands consciousness, but decreases thalamocortical fMRI signals and cerebral blood flow. So I interpret this that membrane-dependent cognition requires energy, and that consciousness might depend on activity going on inside neurons that are low energy, that are quantum processes, which are very low energy. So this fits with an approach that consciousness comes from inside neurons at the quantum level, which we'll come to later. So, but it raises the question, what type of neural activity correlates with consciousness? So if we go back to our third three waves through the brain, conscious perception involves three waves. And the first and second wave, as I mentioned, continued during anesthesia. This is how we monitor evoked potentials in brain monitoring. So 1 and 2 are still there. 1 and 2, first and second waves are not conscious. Only the third wave feedback because it's going backward, it's going in all directions, but it's feedback in this sense, is inhibited by anesthesia, by all three types basically of anesthesia: propofol, volatile inhalational gas anesthetics, and ketamine, selectively preventing consciousness. This study was done by George Mashour's group, University of Michigan, 2013. And George is the department head now, and is a big aficionado of consciousness, does some amazing consciousness research. And I've known him a long time. He comes to our conferences and is actually co-sponsor of our conferences. They've got a big Center for Consciousness Science at University of Michigan that's very well funded. So he showed that only the third wave is inhibited by anesthesia, by not just gas, but propofol and ketamine. So this third wave correlates with consciousness and occurs several hundred milliseconds after sensory input. So you remember from the Libet study that it took hundreds of milliseconds for neuronal adequacy, even if you perceive consciousness earlier at 100 milliseconds. So there's the bing at several hundred milliseconds after a sensory impingement. So here's the conundrum. Activity correlating with conscious perception occurs at 300 milliseconds, but we often respond seemingly consciously within 100 milliseconds. Consciousness is thus deemed an illusion. This is why mainstream neuroscientists and philosophers say that consciousness is an epiphenomenal illusion, and we have a false sense that we're in control. So if we're interacting with somebody rapidly back and forth, and Allison says something, I respond right back, if you measure my brain for the activity that correlates with what she said, it occurs after I've already responded. So the mainstream would say, I'm responding with my inner zombie or my non-conscious autopilot, whatever you want to call it, and have this false illusion that I'm in conscious control. So that's the situation. Consciousness is deemed an illusion. But if we go back to Libet and the backward time effect, it could be that this backward time effect is actually allowing us to be conscious in real time. And it's not illusion, but it requires this trick of backward time effect. And I actually wrote a paper about this called, oh, there it is, How Quantum Brain Biology Can Rescue Conscious Free Will. If you just Google that, you'll find it. It's in Frontiers. And it's actually my most popular paper, 55,000 hits or something like that. And I'm now working with Roger Penrose on an update of that because he has a new take on the physics that allows backward time effects. And this has been reported in many contexts and in the parapsychology literature and even in mainstream neuroscience, but they tend to hide it. They can't explain it, so they kind of ignore it. Okay, this three-wave idea is also consistent with the major theories of consciousness. And the Templeton World Charity Foundation, in their program Accelerating Research in Consciousness, identified 5 major theories. I'm not going to go into this too much. Global neuronal workspace, higher order thought, predictive coding, recurrent processing, integrated information theory, and ORC-OR. ORC-OR is the theory I have with Roger Penrose. And it kind of applies to, the others are all kind of wiring diagrams. Global neuronal workspace and higher order thought both say that the frontal cortex projects broadcasts throughout the brain. So that's kind of wave 3. PC, predictive coding recurrent process, could be in the 3 feeding back against 2. So it's this kind of rubbing against each other, or predictive coding. You have a prediction and then you update it. So that would be PCRT. IIT predicts a projection from the front to the posterior hot zones. And our theory could be compatible with any or all of them. They could all be right or wrong. It doesn't matter. Consciousness still would depend on what's going on at a deeper level. And where that might occur, I'll come to in a minute. So why is third wave feedback associated with consciousness and inhibited by anesthesia? Well, if we get to the cortex has 6 layers, 1, 2, 3, 4, 5, 6. The third wave comes into layer 4. 4 projects to 1, 2, 3, and 6. 1, 2, 3, and 6 then converge on layer 5 pyramidal cells. These layer 5 pyramidal cells have these giant cone-shaped bodies. Their apical dendrites go to the surface and are parallel, perpendicular to the surface, parallel to each other. And this is where EEG comes in. and their motor outputs go to the spinal pyramidal tracts to control behavior. They also have these basilar dendrites that connect to each other, dendritic connections, and this dendritic web goes throughout the entire cortex. And Carl Pribram ascribed consciousness to this dendritic web in pyramidal cells, and I think that may be right. He thought it was causing interference in a hologram that was projected to the brain. But anyway, dendritic interactions in layer 5 may be the origin of consciousness. Bing, bing, bing. So if we look inside a pyramidal cell, here's the apical dendrite that goes to the surface. Here's the axon, and we see the microtubules inside. The microtubules and the axons are different. They're uninterrupted, whereas every dendrites and soma, they're interrupted. And these are the pyramidal outflow tracts to control behavior. I think these are the most likely sites for consciousness in the brain. But they're characterized as Hodgkin-Huxley integrate and fire neurons, as are all neurons. So Hodgkin-Huxley in the 50s developed this idea based on ion fluxes, where there's integration in the cell body and dendrites, which reaches a threshold for firing. So integrate, fire, integrate, fire, integrate, fire, based on a threshold. So this characterizes neurons as threshold logic devices, which you can make a computer of. And so this lends itself to computer analogies. And here we see a bunch of neurons, and here we see a computer node network. And these are thought to be essentially the same. But this makes the system algorithmic, deterministic, machine-like, and too late, really, for consciousness, as I said before. There's no room for consciousness, creativity, intuition, insight, or free will. This is what the Hodgkin-Huxley would predict. Integration here, firing here, or schematically it's here. So it's integrate. It doesn't necessarily have to be linear, but you reach this very narrow threshold, and there's firing here within a very narrow time window. And the firing is thought to occur by sequential openings of ion channels in the axon, which is why it's slanted. However, if you look at an actual pyramidal cell, the pyramidal cells that I've been talking about in awake animals and awake cats, and this was done in this paper by Nandorf et al., you get a different picture. You get a very wide threshold and a very wide temporal variability. And the spikes are vertical. So we have a wide threshold, wide temporal variability, and vertical spikes. The vertical spikes suggest that the ion channels open simultaneously rather than sequentially. But the point is that some non-computable effect, something other than the threshold and the integrate and fire, is regulating regulating the axonal firings and spikes, which control our behavior, control our actions. This is a extremely convenient locus for consciousness to supervene and control our actions by modulating the firing of Hodgkin-Huxley neurons. So there's the Bing. This is where consciousness could regulate behavior, and without consciousness, we can still go about our autonomic, non-conscious activities. This is becoming from a deeper level, and for example, it could be coming from inside the neuron. So here we see a neuron. And the question is, does the variability come from inside, or does consciousness come from inside, or does it come from, as most people say, from membrane-based synaptic activities here at the synapse, or does it come from inside the dendrites and the soma in the microtubules and the cytoskeleton? So I just mentioned the theory I have with Roger Penrose. There's an activity goes on in all the microtubules. You reach a threshold, this threshold, which is the Heisenberg uncertainty principle. There's a quantum state reduction that causes collapse. I'm not going to go into that, but the point is that there's at least one theory, and actually there's others that derive from microtubules and processes inside neurons causing consciousness that could modulate the firing threshold and break us away from being zombies and having no free will and no conscious creativity. So how do we decide between membrane-based receptors mediating consciousness and cytoskeleton or something else? And the answer is anesthesia. What mediates anesthesia? Is it mediated by synapses, which most people would say receptors, or does it come from the microtubules? So in 1846, Claude Bernard showed that cytoplasmic streaming in amoeboid cells, slime mold amoeboid cells, was reversibly inhibited by chloroform. So here's the slime mold amoeba, and this is their cytoplasm, and it kind of flows and undulates in an appropriate direction, actually goal-oriented. And when he exposed it to chloroform, it stopped moving. And when he took away the chloroform, replaced it with air or oxygen, they started moving again. In the 30s, Seifert and Christian showed that this anesthetic effect was directly on the cytoplasm, was not mediated by membrane signaling. So it was happening directly on the cytoplasm and cytoskeleton. And amoeboid movement is similar to how axons and dendrites grow by microtubules polymerizing and then acting at the leading edge. So this process was inhibited in the slime mold. Now, slime mold can actually, amoeba slime mold, they can actually do clever things. And recently, there have been a number of studies where amoebas solve the traveling salesman problem. In computer science, there's this thing called a traveling salesman problem. How do you get, let's say you got to go to 8 cities, what's the most efficient way to go? And what they did was they had an amoeba with 64 little corners that they could flow into, and they would pick 8 out of the 64 and put food in them, and the amoeba would solve the problem by getting the food the fastest by flowing into the proper channels. And so this has been published a number of times, that an amoeba using its cytoskeleton, its microtubules and actin, can solve a traveling salesman problem. Okay, we know that anesthesia selectively blocks consciousness, sparing non-conscious brain activities. What do they actually do in the brain? So anesthetic gases are chemically diverse. They include ethers, halogenated hydrocarbons, nitrous oxide, and the inert element xenon. They don't look like each other. This is very unusual. Usually one class of drugs is like the other components of the class. But what the gases do have in common is that they have outer shells that are filled with electrons. So xenon is a noble gas, so its outer shell has 8 electrons. Anything with a halogen in it has completed outer shells, and nitrous oxide as well. So what they have in common is a completed outer shell without an electron to donate or a hole to receive. So they're basically inert chemically, pretty much. In fact, the more inert they are, the better they are in terms of metabolism, avoiding metabolism, and therefore avoiding toxicity. We heard about sevoflurane possible toxicity a few weeks ago from Phil Malin. So they do share this filled outer shell, which gives them an inert surface. Scientists sought a common feature related to anesthetic potency, 1/Mac, and looked at solubility. So if you look at the human body or any animal body and imagine grinding it up and looking at different compartments where different drugs would dissolve, and this is kind of how pharmacologists think. You get a bunch of different solubility phases listed over here, numbered over here, and here's where they are. And this graph, basically, the key thing is polarity goes in this direction. So here's water, it's polar. And moving in this direction is more polar. As you go in this direction, you get non-polar, lipid-like, fat-like, aromatic rings, and aliphatics. And what they found was, and what they, meaning Meyer and Overton, we've all heard of the Meyer-Overton correlation, Hans Meyer in 1897 and Charles Overton in 1901, they determined the potency of all these gases in mice and salamanders and what have you. And Mac wasn't around then, but it's expressed here in Mac. And they plotted the potency of the anesthetic. So the lower the Mac, the more potent, versus their solubility in olive oil. And this is after searching a whole bunch of different solvents. They found olive oil. And what they found was the potency correlated linearly, almost perfectly, over many, many orders of magnitude. This is really astounding. Methoxyflurane, the MAC is about 0.25%. So this is 1%. This is an atmosphere. So chloroform is about 1. Halothane, I think, is 0.76. Methoxyflurane, 0.25. and you go up, you get less potent, and then nitrous oxide, you know, you need more than 100%. And even nitrogen at many atmospheres is anesthetic, but you have to be in a hyperbaric chamber. You have other problems with that. So they found this amazing correlation over many orders of magnitude. Now, there are a couple of gases, non-anesthetics are called, for example, trifluoromethylbenzene and F6, which follow Meyer-Overton and bind in lipid-like pi resonance regions, but do not cause anesthesia, loss of consciousness. They're called non-anesthetics or non-immobilizers, if you don't believe in consciousness. And this is also from Eger's group. He did a lot of great work. I kind of gave him a hard time about a couple things, but this is really important. And if we can figure out, if somebody can figure out how these gases differ from anesthetics, because they all follow Meyer-Overton, that would be a big clue to understanding consciousness. So going back to Meyer-Overton and the solubility phase is where anesthetics act, where consciousness should arise is in these aromatic rings, xylene, toluene, benzene, down here, highly nonpolar, lipid-like. So benzene is the basis for organic chemistry. We all studied it. It has 3 extra electrons in a 6-carbon ring. The 3 extra electrons are represented like this, and they form an electron cloud above and below the ring. And these electron clouds are nonpolar and inert, but they can have dipoles and they can induce dipoles. So if you have 2 of these rings, they're attracted by van der Waals attraction, and this one induces a dipole in this one, and this one induces a dipole in this one, and they oscillate back and forth. I should say that benzene in bulk form in your gas tank is explosive, but benzene, when it's in a geometric array and spatially separated, for example, in aromatic rings and proteins, has a lot of very interesting quantum properties. For example, graphene is, if you can see, these are all these hexagons. It's just a big sheet of benzene put in one layer. And it has interesting quantum and electronic properties. And when you put 2 of these sheets overlaid each other, so here, and tilt them, angle them at just the right angle, 1.1 degrees, for example, 2 things happen. You get superconductivity, it gets to be a quantum system, and you get these moire patterns where you can see larger hexagons made up of smaller hexagons. And this is kind of like a hologram, which Carl Pribram suggested might be mediating consciousness. But it just shows you that benzene and graphene has a lot of interesting quantum properties. And these rings coalesce in nonpolar regions inside proteins and lipid membranes. So in proteins, we have aromatic amino acids that are in the middle and are called hydrophobic pockets, for example, first in micelles and then proteins. And lipid bilayers, they get together in the middle, and the aromatic rings, the benzene type rings, are in the middle. And these are the nonpolar lipid-like regions where anesthetics bind. I should also say that the same pi resonance rings are found in psychoactive and psychedelic molecules. Dopamine and serotonin have them. Serotonin has the indole ring with a 6 and a 5. And the psychedelics all have them. LSD has a extended conjugated ring. DMT has the indole ring, as does psilocybin, which caused those effects in Carhart-Harris's study. So they have something to do with psychoactivity and mental states. In proteins, the aromatic rings, tryptophan, phenylalanine, are found inside proteins. For example, tubulin, that has 86 of them. And these little spheres, which you might be able to see, are where anesthetics bind. We kind of simplified this to show a dipole, that these all could be aligned in a dipole in a little model that we have here. I'll come back to that. So they can align and oscillate and form dipoles that wrap around the microtubule. This is part of our theory that I'm not going to really talk about too much. Now, the anesthetic gases, as I mentioned before, have regions that are nonpolar with completed outer shells, and this makes them able to bind, form their own van der Waals forces with aromatic rings and other nonpolar groups. So basically, This is kind of the punchline, although I'm going to keep going for a while. But if you have aromatic rings, they could be part of amino acids inside a protein. They're going to oscillate in terahertz. And what anesthetics do is they come in and they form their own van der Waals forces, but they're called dipole dispersion forces because they don't match up quite the same, and they block the oscillation. So I think what's happening is that anesthetics prevent dipole oscillations among these aromatic rings somewhere in the brain. So this is anesthetized unconscious on the right. This would be conscious or potentially conscious on the left. So for most of the 20th century, going into the 20th century, scientists knew that anesthetic through Meyer-Overton bound in nonpolar lipid-like regions. And the first attempt at explaining anesthetics was that they get into the lipid region of the membrane and just by bulk volume expansion cause prevent activity just due to volume expansion. Critical volume hypothesis was the first theory. And then in the 60s and 70s, it became realized that proteins, receptors and ion channels in membranes were essential for neuronal membrane signaling and Hodgkin-Huxley behavior. And Jim Trudell, whom I knew, and Bruce McIver was a good collaborator of him, wanted to keep the lipid site of action. So he used a sodium channel, which would open. And he said the anesthetics get in and caused a phase transition in the lipid, and that extrinsically prevented the ion channel from opening. So he was incorporating ion channels, but still relying on lipid site of action. But the problem was that 2-degree temperature shift had the same effect as the phase transition that he was purporting was causing the anesthesia, and you don't lose consciousness with a slight fever. So this turned out to be not correct. And then in the 80s, Nick Franks and Bill Lieb showed anesthetics act directly in proteins in nonpolar hydrophobic pockets, for example, in the membrane-free luciferase. So inside luciferase, and they could measure its activity by light emission. And this is essentially firefly protein. And they found a inhibition of the light emission from firefly protein proportional to MAC. So they show that you don't need lipids, you don't need membranes. You can act directly on proteins. So which proteins? Well, the obvious choice to most people was ligand-gated ion channels. And indeed, anesthetic gases do bind to membrane receptors for serotonin, glycine, acetylcholine, and GABA-A being an inhibitory neurotransmitter. But experiments produce conflicting results. Some anesthetics open a channel, others close it. Not all anesthetics bind to any one receptor. In an editorial in anesthesiology, Alex Evers and Jim Steinbach called anesthetics double-edged swords, saying that volatile anesthetics both enhance and inhibit ligand-gated ion channels. Well, if they enhance the inhibitory and inhibited the excitatory, that would work, but that's not how it was. The volatile anesthetics both enhance and inhibit excitatory acetylcholine receptors, and they enhance the inhibitory at low dose, but inhibit at high dose. So this is the opposite of what you might expect. A number of other papers showed that anesthetics do not act on membrane protein receptors, despite what everybody says. At the top, acetylcholine, no longer in the picture. Acetylcholine 5-HT, opioid, adrenergic, and potassium receptors do not mediate immobility, which is the code word for anesthesia if you don't believe in consciousness. GABA receptors do not mediate the mobility. GABA receptors may not be involved. Genetic inactivation GABA receptors had no effect on immobility. Despite this, I'm pretty sure the textbooks still say GABA receptors. And Ted Eager, another really good thing that he did in a paper in 2008, realized that this wasn't working and kind of waved the white flag and said that is a new paradigm needed to explain inhaled anesthetics, how inhaled anesthetics produce immobility. You can read it. 2 decades of focused investigations have not identified a channel that alone is sufficient to mediate immobility. He named all the channels. Furthermore, no combination of any of them seemed sufficient. And he called for a new paradigm. When I read this, I said, yeah, microtubules. And I wrote to him, I wrote to the editors and suggested this, but Eager suggested going back to lipid theories, which have gone nowhere in the last 13 years. So a new paradigm is needed, but not the one he selected. So anyway, the field was at a crossroads. Go to membrane proteins despite these negative findings, or go back to Claude Bernard and the cytoskeleton. So we mentioned Claude Bernard, and in 1968, Allison and John Nunn, shown here, who was from the UK and whom I met several times. He was a friend of Brunel's and came to visit and give talks several times. Did a study on actinosphaerium, this little urchin, which had these axonemes sticking out. The axonemes were made of microtubules in this double spiral. So this is a cross section of one of these guys. And when they exposed them to halothane, all the axonemes depolymerized. They just vanished. They just disassembled. However, this was at 5 Mac. But it showed that anesthesia does bind to tubulin and microtubules, but depolymerization was not the cause of anesthesia. However, it makes you worry because depolymerization of microtubules is basically Alzheimer's disease, or can cause something similar to Alzheimer's disease, and too much anesthesia over a prolonged period of time or repetitively might be a problem. Brunel knew I was interested in microtubules and consciousness and handed me this paper and said, if you want to figure out anesthesia, figure out consciousness, figure out how anesthesia works, and you knew I was interested in microtubules. So this helped convince me to go in anesthesia, even though it was at 5 Mac, which, so depolymerization per se does not cause loss of consciousness. Now, more recently, well, in 2008, Eckenhoff's lab, Rod Eckenhoff, shown upper left there, and his group at Penn did a systematic approach. They found 70 proteins. They did radio-labeled halothane binding in mouse brain. And they found 70 proteins in the brain neurons misspelled there, sorry, found to bind anesthetics, half in the membrane and half in cytoplasm. They then did genomic, they looked at altered genetic expression of certain proteins, and they found 3 proteins common that were whose genetic expression was altered by both anesthetics, tubulin and 2 other proteins which don't seem relevant to any signaling. And so among the candidates, they concluded that proteomics and genomics point to microtubules as the site of action of anesthetics. They also showed, by the way, clinically that people on Taxol, like for breast cancer, which stabilizes microtubules, require more anesthesia. Their MAC is higher, possibly because the microtubules are harder to get to. They then did a study with tadpoles, and tadpoles have conveniently transparent heads, and they used a fluorescent anthracene anesthetic, which they gave to the tadpoles, which was only active as an anesthetic when they illuminated it with ultraviolet light. So they gave them this anesthetic. They're swimming around, they turn on the light, and they just stop and roll over, and they're anesthetized. And they then donated their tadpole brains to science, and they found that the anthracene was bound to microtubules. So they concluded that direct modulation of microtubules contributes to general anesthesia. They wouldn't say that it's the end all be all, but they gave us a shout consistent with our theory, although that's not quite our theory, but close enough, that microtubules are responsible for consciousness. Now we meaning my colleagues, Travis Craddock, Jack Tusinski, and others, in this paper in Nature Scientific Reports in 2017, did a computer modeling study. And this guy, Philip Kurian, had access to this gigantic computer somewhere back east. And I showed you the oscillations of two benzenes. So what they did was take all 86 pi resonance rings and model the oscillations and interactions between all 86 of them at room temperature, and using these 3 aromatic amino acids, tryptophan, phenylalanine, and tyrosine. And what we found was at ambient temperature at KT, a spectrum of collective dipole oscillations with a common mode peak at about 613 terahertz in the blue light region of the spectrum. The presence of each of 8 anesthetic gases and non-anesthetic. So there's methoxyfluorine, halothane, isoflurane, enflurane, sevoflurane, diethyl ether, and desflurane, and nitrous oxide. The presence of each of the anesthetics abolished the 613 peak and dampened other terahertz frequencies proportional to anesthetic potency. So here's some subtraction spectra. So here's the 613 terahertz peak with desflurane, and negative means it's gone. So it's missing from the spectrum that had them. The same with diethyl ether, 613 is gone, enflurane, halothane, isoflurane, methoxyflurane, nitrous oxide, SIVO. Now, F6 is one of those non-anesthetics, okay? It follows Meyer-Overton, but it doesn't cause anesthesia, and it did not affect the 613 peak. It affected another peak that was lower. TFMP, trifluoromethylbenzene, actually increased the 613 terahertz peak. And fluithol, which is a convulsant, an anesthetic and a convulsant, acted as an anesthetic and diminished the 613 peak. So all the anesthetics abolished the 613 terahertz peak. The non-anesthetics did not. Now, the 613, just by the way, is in the blue or blue-green spectrum. And in Hindu philosophy, the Hindu deities are often portrayed as blue when they're being spiritual. So that may be a coincidence, I don't know. But let's go back to the non-anesthetic gases, the TFMP and the F6, which follow Meyer-Overton, but do not cause anesthesia. And here we see them over on the right, where their MAC would be 1,000 or could be infinite because they don't cause anesthesia, and where their solubility would be. And so they're in the range of where anesthetics should act solubility-wise. They're between halothane and methoxyflurane or down by sevoflurane. Why don't they cause anesthesia? So this is the oral gas partition coefficient. This is MAC. So as I said before, they don't abolish 613 peak. They abolish a lower one or they increase it. Why is that? They had no effect on tubulant terahertz. Why not? Well, we plotted all the anesthetics and non-anesthetics against another property, polarizability. And what's interesting before I get to the non-anesthetics is that all the anesthetics, this is methoxyflurane, this is diethyl ether, you get a pretty good Meyer-Overton-like correlation just for polarizability. So this might have something to do, and this has to do with how the dipoles can oscillate. And the non-anesthetics are out of range. They're either way high or just above, or they're actually too potent. So they would be more potent than methoxyflurane. So what we think this meant was that normally the dipoles are oscillating like this. An anesthetic is going to gum up the works and prevent dipole dispersion, but the non-anesthetics go along for the ride. They're highly polarizable. So when these guys polarize this way, they go along for it and they oscillate back and forth. They apparently just go along for the ride due to their high polarizability. And even though they're binding where the anesthetics bind, they don't cause anesthesia. I think this is the only explanation for non-anesthetics not causing loss of consciousness. And this is part of our overall theory. I'm going to wrap up now of a multi-scale hierarchy where you start with neurons. Instead of going upward, well, in addition to going upward, you go downward in scale. And we know that these oscillations occur at these levels in microtubules and the tubulins. And down here with the pi resonance cloud dipole oscillations is where anesthetics act to gum up the works. So therefore that blocks everything going up higher. And down here it connects to the physics of the universe, but that's another story that with Roger Penrose's work and so forth. And here's some evidence for these coherent resonances from Anirban Bandipati's group at levels of neurons, individual microtubules, and tubulins, where you see terahertz, gigahertz, megahertz, kilohertz oscillations over many orders of repeating every 3 orders of magnitude in this triplet of triplet arrangement. So these are repeating cell-similar fractal-like patterns that repeat all the way up from at least from here all the way up to neurons. Conclusion: despite conventional wisdom, objective evidence points to the origin of consciousness in the site of action of anesthetic gases as quantum vibrations in microtubules inside brain neurons. And by the way, if you go to Wikipedia and look up theories of anesthesia or theoretical mechanisms of anesthesia, go all the way to the bottom. There's a very nice section on the quantum vibration theory of microtubules mediating anesthesia. And finally, this hypothesis is currently being tested in the TWF Templeton World Charity Foundation program and experiments at Princeton and South Florida. And let me just say one thing about that. Photosynthesis proteins were the first proteins to show quantum states at warm temperature. Everybody said it's you have to be really cold temperature. And what you do to show it in a protein is shine a laser, several lasers actually, and you measure signals. And if you get the sawtooth peak, that's indicative of interference patterns. So this was shown for photosynthesis protein, which is what proved that they have quantum coherence. And we are in the process of doing that exact same experiment or similar experiments in tubulin at Greg Scholl's lab at Princeton, even as we speak. And Greg Scholl is a leader in quantum biology, and he's not an aficionado of microtubules or anything else, but we've already found significant quantum states of microtubules that last 5 nanoseconds, which we think is the world record. So we're currently being tested. We've shown the quantum states. We then intend to study effects of anesthesia on them. And if we get a Mach-like Meyer-Overton correlation with anesthetics dampening the interference patterns and other quantum states in microtubules, I think we'll justifiably be able to claim that anesthesia acts by dampening these vibrations and quantum vibrations in microtubules, which are also the origin of consciousness. Thank you very much for your attention. I'll be glad to take some questions.
Oh, that was all really fascinating. And I think I'm going to need some time to keep thinking about it and let it soak in before I have near enough understanding as I should. But one question I did have was a lot of the work that you mentioned on the tubulin terahertz was on inhaled anesthetics. So how do you address IV anesthetics?
Right, great question. We're going to do propofol, and I should say off the bat that propofol, ketamine, and etomidate all do bind to microtubules, to tubulin, and they get in and they get inside the cell. I mean, supposedly the ketamine acts at NMDA receptors and propofol acts at GABA receptors, which they undoubtedly do, but so do the volatiles. They all act at the membrane proteins. But that doesn't mean they act there. You know, anesthetic gases go to fat stores, lipid stores. So I sometimes tell the residents there's more anesthetic gas in the patient's butt than in their brain, but you know, the anesthetic's working in the brain, not in the butt. So there may be anesthetic gases in the membrane receptors, and there may be propofol in the GABA receptor. There is, we know that. But that's not to say that's where they act. They're going to go to any nonpolar region. And we've done some studies on propofol binding to tubulin, and we will test propofol. Bruce McIver, who's doing the anesthesia experiments, who is not a microtubule person, he's a GABA person. He's GABA A receptor. He believes that it's GABA A receptors, and he was going to. I invited him to this talk, but he's in Hawaii, so he's going to watch the replay. And so he asked me to say, I said, yeah, let's test for propofol. And propofol, if you look at the molecule, it's basically a benzene ring with a couple of doodads on it. It's very much like the other, and etomidate and ketamine also. They all have these pi resonance rings. And therefore, they're likely to get inside the neuron in addition to binding the receptors. So that's a very good question, and we're going to test that. But it's unclear whether they follow Meyer-Overton or not. But nonetheless, if they cause loss of consciousness, and our theory is right, they should directly or indirectly dampen the quantum vibrations in microtubules. So we will definitely look at that.
Very cool. Thank you.
Stu, can you talk about memory and how this plays out with memory a little bit in maybe a minute or so?
Yeah. So most people say that memories and synaptic plasticity, you know, the long-term potentiation, if you stimulate a neuron high intensity briefly, you get a prolonged sensitivity in the postsynaptic neuron. So the receptors, synapse adapts and becomes more sensitive to whatever caused the initial one. So you can elicit memories. That's a theory. The problem is that those membrane proteins are in the synapses are transient. They're recycled hours to days. They're only there a short time, and yet memories can last lifetimes. So we think that memory is in microtubules. I know it sounds like I sound like a broken record, but there's more. There's actually some good indication for that. If your microtubules fall apart, you get Alzheimer's. That's number one. I didn't show this, but we did a study a few years ago with a modeling study with calcium calmodulin kinase 2, CAMK2. And in long-term potentiation, calcium comes in and activates CAMK2 and this hexagonal kinase domain. And I remember looking at this hexagonal kinase domain, and I remember Brenda Kent was talking to me in the background, and I was kind of zooming in and on this, and unfortunately ignoring her, which she reminded me of much later. But the hexagon of the CAMK2 precisely matches the hexagon of the tubulin lattice, microtubule lattice. And so we did computer modeling, and we showed that the CAMK2 can bind to microtubule lattices and phosphorylate up to 6 tubulins at a time, 6 bits of information at a time. And thousands of these things come in with each synaptic event. So I would argue, and I would bet money, that memory is in microtubules. There's each tubulin in a microtubule lattice can be post-translationally modified, phosphorylated. Many things can be done to program it, to encode it. So it's really the perfect system to encode memory. And the microtubules in dendrites and soma are stable. In non-neuronal cells, they have to depolymerize and then repolymerize for mitosis for cell division. In neurons, they don't divide, so they don't have to do that. And in dendrites, they're capped at both ends, so they stay the same. So you can encode a long-term memory in microtubules and perhaps neurofilaments, which are even more stable. So in a nutshell, memory in microtubules.
Hey, Dr. Hameroff, I was just wondering, do you think that you have to be able to form memories in order to be conscious? Like, is that a part of the definition of consciousness? For example, when a patient wakes up from anesthesia and they had a conversation with you and they asked the same question in 30 seconds, could it be said that they're in that zombie mode type of thing? Or do you need to have that to meet the definition of a conscious being, or however you would classify it?
Right, that's an excellent question. And let me first say that some people claim that that our patients never lose consciousness; they just don't remember. And you know, we're torturing them; they're being operated on, and they just don't remember.
We call those.
And it's hard to refute that because we can't prove they're conscious. I can't prove that anybody's conscious. You can't prove that I'm conscious. All I know is that I'm conscious, and I assume you are. I don't. But in answer to your specific question, you don't need memory to be conscious. And there are people who are amnestic. HH, a famous guy who I forget what part of his brain he lost, but he had he had total amnesia. He was conscious. So I don't think you need memory to be conscious. But I don't think that anesthesia is loss of memory with consciousness, because you know, if a patient gets light, they're autonomic, they're going to see autonomic responses, et cetera, et cetera. But it's impossible to refute that because we can't prove consciousness per se.
So piggybacking off of that, how could you possibly prove consciousness, or is that a talk for another time?
How could you what?
Oh, sorry. How is there a way to prove that someone's consciousness? Like, do you think like within the next 50 years we'll be able to prove that someone's conscious and someone's not, like pending their political party or something like that? I don't know.
Please don't bring politics into this. Maybe. In the Templeton project, one of my adversaries said, wanted us to do what we're attempting to do at the Princeton lab on individual tubulins in a brain in a moving monkey. And I said, not yet. Maybe in 50 years we'll be able to do that. It's possible to. So the point is that if we could show these quantum interference, assume we can show it in tubulin and microtubules. If we could show it in a brain of a living entity, then maybe. But according to our theory, you also need to reach collapse. You need to reach this quantum state reduction, which makes it a little bit trickier. So eventually, maybe, but I think we got a lot of work to do first before we can even answer that question. Find out exactly where it's coming from and how anesthesia works. So it's all, but it's also, people talk about downloading or uploading their consciousness. Billionaires want to be able to put their consciousness in a computer when their body dies and so forth. So there's all this effort. So AI, it's kind of a big scam. People either preserving their brains and cryopreserving their brains, freezing them, which destroys all the microtubules, by the way, or downloading into some kind of computer. But classical computers, I don't think will ever be conscious. The only possible way might be to use some biological system like graphene, like I said, or fullerenes, which are nanotubes, which have these pi resonance. Something like that. It might be possible to have be able to download your consciousness into something like that. Maybe, I don't know. That would be the only way. I don't think putting in a classical computer or preserving brains could do it.
Thank you, sir. That was a great talk.
Thank you.
Hey, Stuart, it's Phil. Can you hear me?
Hey, Phil, how you doing?
Do microtubules communicate across cells?
Oh, that's a good question. Yes, and I didn't show my friend Anirban Bandyopadhyay, who's I mentioned his other work, has a paper coming out in Journal of Neurophysiology. This is really interesting. He basically shows, well, he uses something called a dielectric resonance probe. That's kind of like an ATM tip that goes inside the cell and can measure 3 different frequency ranges, or 2 different, gigahertz and megahertz. And he finds gigahertz and megahertz oscillations in a living cell with this probe. And then he correlates the frequencies in one area of the cell and another area of the cell, or in one cell and another cell. And he has found correlations. It's just kind of, and he finds that they're coming from, and then when he gives drugs that wipes out all the microtubules and all the actin, and there's no cytoskeleton, there's no correlation. But interestingly, the axon still fires, the membranes still fire, but we know axons fire without consciousness. We know membranes fire without consciousness. So that's consistent with what I'm saying. But as far as your question, there's the ephaptic transmission in the brain, which is basically electromagnetic field, pervade the brain. And that's one way that there can be interaction among neurons that don't require synapses. And how that electromagnetic field is mediated is another question. Anirban's work would suggest they come from the cytoskeleton, which are able to do stuff like that. So interestingly, in Anirban's paper, he showed that the microtubules and the actin were causing this correlation, and also affecting those in the dendrites. So the axon goes in, it branches, and it turns out that this branch can fire and this one doesn't. So there can be selective firing in the distal arborizations of the axon. I didn't know this. And apparently activity in the soma and the dendrite can determine whether it's this axon branch that fires or this axon branch that fires. And there's this non-local correlation, and even activity in another neuron can do that. So but it showed that the microtubules are responsible. But Anirban said the editors of Journal of Neurophysiology, he followed them for 3 years to get this in print because it's so contrary to conventional thinking. They finally agreed, but they said, okay, but you can't mention Hameroff or Penrose, and you can't mention microtubules. So he didn't, although he said filaments. He called them filaments, and then somewhere in a little footnote, it's filaments are microtubules and actin and filaments. So I guess I've pissed off a lot of people in the scientific world, but they think we're crazy. But the data's on our side. I'm sorry, and we'll see. So this paper will be coming out in Journal of Neurophysiology, even if it doesn't mention us. it supports exactly what we're saying. And he has another one come out in a different journal along similar lines. So the answer to your question is yes.
Great talk, Dr. Hameroff. Other than avoiding anesthesia and being otherwise healthy, what can we do to safeguard our?
Safeguard, like you mean like for Alzheimer's? For is that what you're referring to? I can't see who's talking, but you know, I worry about elderly patients, also peds. Allison probably knows way more about this. But kids who get multiple anesthetics, and it doesn't matter whether it's gas, propofol, ketamine, they can get, of course, there's so many other factors just being in the hospital and so forth, but they're seeing, and so this is a controversial subject, and anesthesiologists sometimes like to kind of hide this, that there could be some deleterious effect of repetitive anesthetics in babies or in elderly. But I think, and I wrote an article about this recently, and you know, you want to avoid prolonged, you want to avoid repetitive, and you want to avoid cold temperature, I would say, if you're because cold destabilizes the cytoskeleton. So if you have to repetitively anesthetize anybody, but particularly babies and elderly, keep them warm and try to, you know, minimize the MAC. I don't know whether mixing propofol and gas, you know, keeping low levels of both would help. I don't know. The data on this is very murky, but I think there's a real effect on, and it's probably mediated through the cytoskeleton, through the microtubules, since, as we saw, 5-MAC at least depolymerizes microtubules, at least in a sea urchin, and may do something similar in our brains.
Yeah, and there's some evidence that drugs like dexmedetomidine and xenon have some protective effect to the more standard like sevoflurane-induced neurotoxicity that we've seen in animal models and things like that. Although, yeah, exactly, we don't know a lot of information about in clinical studies with prolonged or multiple anesthetics yet in children. Those are.
Yeah, thank you, Alison. And Mervyn Mays has been pushing that for a while. Is there any evidence that xenon's protective?
Yeah, in some of their animal studies, there's been none in humans yet, I believe. At least has been published. Maybe they're working on it right now, but yeah, both dexmedetomidine and xenon. There's a paper I found a couple years back. I can send you if I can find it again.
Yeah, thank you.
That.
Since we were talking about the kids and anesthetic toxicity, what about inflammation in the brain destabilizing the microtubules? Isn't that I mean, wouldn't that be more of a concern than even the anesthetics?
Yeah, inflammation it seems to be correlated with dementia and Alzheimer's and so forth. But you know, I often wonder, dementia inflammation can destabilize microtubules, but destabilized microtubules might also cause inflammation. For example, tau. We know about tau as a marker of Alzheimer's and brain damage. And tau is actually a microtubule-associated protein. Its normal function is it's stuck on a microtubule in a specific location on the microtubule lattice. And in fact, there's evidence that the precise location is a signal for trafficking. You know, materials that are required for a synapse, for upregulated or downregulated synapse, are synthesized in the cell body and then transported along the microtubules by these motor proteins that quite literally walk along, or actually run along the microtubule carrying an enzyme. And paper, I didn't show the slide, but somebody showed that the tau protein tells the motor protein where to get off. So when you think about it, these motor proteins running along these microtubules, particularly dendrites, so the microtubules are interrupted. They have to go along, they have to jump from one microtubule to another. Then they come to a branch point, you know, in dendrites. Do they go this way or do they go that way? And then they come to the right synapse and they have to say, okay, get off here and deliver your cargo to this particular synapse, which needs upregulation for this enzyme. And, you know, how do they know where to go? Well, it turns out the tau protein is a traffic signal that tells them where to get off and where to deliver their cargo at a particular synapse. So that's synaptic plasticity. Synaptic plasticity depends on the microtubules and the tau proteins as encoding. It's really a form of memory because it's telling which synapse to fortify. The tau is also a marker of inflammation and of demise of dementia and Alzheimer's. And it's unclear whether the tau falls off the microtubule and that destabilizes the microtubule, or the tau gets hyperphosphorylated first and then falls off. It's unclear which comes first, but in both cases, you lose the microtubules and then you lose synapses, and you lose the advantage of the tau on the microtubule. And when that happens, stuff gets released and you can cause inflammation. So we don't know which is the chicken and which is the egg, but I would say we have to consider at least that the microtubules falling apart, release of tau, breakdown of the membrane will release some kind of nasties into the general circulation, which result in inflammation.
Well, thank you so much, Dr. Hameroff. Does anyone have any other questions for him? Very enlightening talk. Thank you so much. And thanks so much. I'm just going to take a couple of minutes to remind everyone about the bedside pass thing that I had presented last week. That's going to go live June 1st. I'm going to share my screen just again to remind you guys what that is. So this is the bedside procedure pass. As I mentioned last week, this is going to roll out June 1st for any procedures that we do outside of the operating room. This includes arterial lines, blocks, neuraxial catheters, anything like that. And the nursing staff will also be trained on this as well and aware. And so essentially every time that we need to do a procedure that is outside of the operating room, this procedure needs to, this pass needs to happen. So you need to have a briefing sometime beforehand with the team members involved. Doesn't necessarily need to be in person. You can do it over the phone. The timeout, of course, as usual, should be done immediately before you either touch the patient or start giving anesthetic for the procedure, and then a debriefing afterwards as well. We had discussed how, in this, our banner leadership had, said that residents or non-attending providers could be performing this part without supervision as long as they were considered to be sort of signed off by their program. And so Paul and I sort of discussed how we were going to define that for everyone. And I think what we felt like would be easiest and most straightforward, instead of giving everyone numbers for each procedure, is that the resident should just communicate with their attending who's going to be officially supervising that procedure with them. Either the night before, if it's a planned thing, or right before, if it's more of an unplanned procedure. And that communication should happen and should be decided by both parties on how much supervision is needed by the attending for each part of this. Okay, so that will just kind of make it simple for everything. So just communicate with your attending. You guys come up with a plan that everyone feels comfortable with. And then whether that's just the resident and the nurse, or the resident and the attending and the nurse, make sure to please do this procedure. And we'll make sure to have copies of this in block carts, NOB, PACU, pre-op, all of those areas by June 1st, so that you can have this on hand. And also, I believe we'll try to get like a PDF copy or something from it as well. So maybe we can put it on the Propofol site, and that way you guys can access it there as well. Are there any questions about that? Or Paul, did you want to add anything?
I'll just say that, it's just a part of the, communication, if you're going to do an A-line in pre-op, talk about it with, that's a part of your plan that you discuss with the attending the night before. And then you just do a timeout with the, with the nurse in pre-op right before you do your arterial line. If you're up in the ICU, and you're going to do a central line where this would be a part of, you'd need to do a timeout. You're not going to do a central line on a patient without, discussing it with the attending and talking about, doing the central line or the arterial line or whatever. So this isn't like, a big new thing for our department. You're not gonna do a block without, calling the block faculty and say, hey, we're in Bay 23, we're ready to do a block. This isn't a big change for our department. This is, we're going to talk about communicate before we do a procedure, and there's going to be a few things that the nurses are going to start adding to help make things more safe for other departments. But our attendings are going to be, for the anesthesia department, are going to be there and be aware of what's happening with the patient. So it's not, this isn't a big shock and surprise, and so. Was that clear, what I said? Just call your attending before you do a procedure.
Yeah, call your training and make sure the nurse is there to go through this with you.
Anybody questions regarding how we're going to handle this? Cool. I'm going to sign off.
All right. Thanks, everybody. Thanks again, Dr. Hameroff.
Thank you.
Yeah, thanks, Stu.
Thanks, everybody.